So welcome guys to the very first F22 final exam review. This is one of a three-part series over the next few days. So Tuesday, Wednesday, Thursday. So please come around Wednesday and Thursday if you can to get the entire materials review. We'll be covering topics 1 through 4 throughout this review session. So that's about 40% of what you should know for the final. But there may be other material outside of these review sessions that you should use as a way to prepare for the exam. We've included the most important stuff, but there are definitely details in class and in other materials that you should go over. A couple of things I'd recommend. I would go over, of course, the exams, but not just what you got wrong on the exams, but also how you answered problems, whether or not you got them correct or not. If you wrote something that got full credit, but you wrote way too many details, you want to make sure you're optimizing your energy that you expend on the exam, because just like other exams, it's pretty long and you don't really want to be wasting any energy on questions when you need to be saving your energy for other questions. While there isn't really like a time limit that's super restrictive to most people, you still aren't gonna have like an unlimited amount of energy to waste on all these questions. So it's best to just see how your answers compare to the answer keys, and it's good to also do that for the problem sets as well. Looking back at your answers versus what was on the answer key and seeing if you could answer those now as well. Because a lot of the stuff, especially the stuff we're covering today, will be from very early on in the semester. The last week that we cover in this goes back to the first week of October. So it's been a while. So I hope you enjoy this trip with us on the sleigh of education into the winter wonderland.
Okay, so we're going to start by talking about just a brief introduction about amino acids and their structure and important groups. Okay, so amino acid structure, it has that central alpha carbon, has an alpha amino group on the left, an alpha carboxyl group, and all amino acids have different R groups. These are what gives the characteristic to every single amino acid. We also have them. Wait.
Sorry.
Okay. All right. So they're also pH ionized dependent for the ionization. So at low pH, we're gonna have a +1 charge. So the carboxyl group is going to be neutral, and the alpha amino group is going to have a +1. The neutral pH for each amino acid is just going to be neutral. So we're going to have the charge for the carboxyl cancel out with the amino, and at high pH is just going to be a negative charge. Okay, now we're going to go over a peptide bond. So peptide bonds formed through a dehydration condensation reaction, meaning that as a result, we get water formed as a product. And peptide bonds are covalent bonds. That's important to know, and that means that they are very strong. And to break up peptide bonds, we're going to need to use a lot of energy, and the reaction that's involved in breaking peptide bond is hydrolysis reaction, and in that reaction, it's opposite to the dehydration, so we need to use water to break the peptide bonds. Okay, so now we're going to just go over the levels of protein structures. So first level, primary, and this is just the sequence of amino acids, and they're all linked together through covalent bonds. And again, that's just gonna, if we need to break it, the primary structure of a protein, we're gonna need to use a lot of energy to do so, since it's covalent bonds, and they're very strong. And then we have the second structure, secondary. And these are interactions between backbone atoms. And for the secondary structure, we have the alpha helices and the beta pleated sheets. And the type of interactions that we have for a secondary structure is a hydrogen bond. And again, this happens only between the backbone atoms of the protein, so it doesn't involve the R groups. And for the orientation for the alpha helix, we have the R groups on the outside, as it shows in the picture up there. And for the pleated sheets, we have them alternating. We have one down, and then the next one would be on the opposite side up. And again, since for secondary structure, the R groups are not involved, that makes them ubiquitous. So that means that they just are in all proteins, since they don't have like specific characteristics, since R groups are not involved. Okay, and then we have the tertiary structures, and these are like forming one polypeptide, and that one involves interactions between R groups. And the types of interactions that we have could be hydrophobic, van der Waals, ionic bonds, hydrogen bonds, and we also could have disulfide bonds or bridges, and these form between cysteine amino acids, between the sulfurs of the cysteine. Okay, and then the quaternary structure, and this is kind of similar, like it has the same type of interactions as the tertiary. The only difference is this one is made up of multiple polypeptides, so it has multiple subunits. And not all proteins have quaternary structures. Some of them do, and some of them don't. And for an example here, we have the hemoglobin. It has multiple heme groups. For each heme group, it's a subunit or a polypeptide. Okay, and then we have a question. What do you guys think would happen if we have a molecule that disrupts hydrogen bonding, such as urea? What structures of like the protein, or what levels would be affected? And Zoom, just feel free to type on the chat, or here in the room, just raise your hand or just yell it out.
Said secondary structure.
Yes, exactly. Secondary structure could be, it would definitely be disrupted. Do you think disruption will happen also for tertiary and quaternary structures? Yeah, it would, because for secondary will definitely be affected, since it's just made up of hydrogen bond between backbone atoms. But we also, for quaternary and tertiary structures, we have hydrogen bonding is one type of interactions that take place. However, the primary level structure is not gonna get affected, since it's covalent bonds. And these are very strong, so they don't involve hydrogen bonding. Okay, and then for just like similarities and difference between quaternary and tertiary structures, as I said, tertiary structure is a single polypeptide chain, and it just falls in a 3D shape. And then quaternary structure is multiple polypeptide chains that are interacting together, so multiple subunits. And again, the type of interactions that take place are all, it has some covalent and non-covalent bonds. The only covalent type of bonds that takes place for these levels is the disulfide bonds or bridges. And then all the other interactions are non-covalent. So we have hydrogen bonds, electrostatic interactions such as ionic bonds, and van der Waals. And then we have a domain versus a subunit. And this has to do with like the tertiary and quaternary. So if we think about a tertiary structure, this only is like, it's made up of domains. It doesn't have subunits. I have an analogy if we think about it from like human wise or a stick figure. One stick figure has multiple domains that are like the hands and the legs. These are domains in one person, one human. But then if we have like 3 friends and they're holding hands, they're interacting together, but each percent of them is just one subunit, and each subunit has domains within itself, like within. So the hands, legs, these are all domains. I hope that helps. Just helps me to think about it that way. Okay, and then now we're gonna just talk about like the R groups and the type of interactions and bonds that take place. So we have polar R-charged groups, and these ones are right here. We're gonna have the positive charge, 3 of them, and the negative charge, 2 of them. And these ones will, if they interact with together, they're gonna make ionic bonds or electrostatic interactions. And then we also have the polar non-charged amino acids right here. And these ones could do hydrogen bonds or other dipole-dipole bonds. And they're hydrophilic because the type of bond that would take place, it could be if it's a hydrogen bond or other dipole-dipole. CO, OH, and NH, these all have like a high difference in electronegativity, and that would make it a polar covalent bond. And if we have 2 like atoms from polar covalent bonds, they could make a dipole-dipole interaction or a hydrogen bond if it's between a hydrogen and a nitrogen, oxygen, or fluorine. And then we have the non-polar R groups, and these ones have a lot of like carbon-carbon interaction or carbon-hydrogen interaction. And these are covalent bonds, but they're non-polar. And the reason because between carbon-carbons in the same molecule, there is not a high difference in electronegativity, and the same goes for carbon-hydrogen. And these form also like London dispersion forces. Okay. And then now I'm just gonna talk about like the Stark hindrance and how it affects like a protein. And so the main idea here that we need to take is that the R group size and charge and like orientation would affect or can affect the structure of the overall protein. And so if you look at glycine here, its R group is just a hydrogen. And so that's very small. And that would make it very easy to be like in the middle of like a polypeptide or the peptide sequence. And then if we look at proline, this one is cyclic and it's very kinky or kinked, and that would like, we wouldn't find it in like, let's say an alpha helix. And if we look at it here, the R group is connected to both the central carbon and the alpha amino group of the amino acid. And then if we also look at the phenylalanine, it's aromatic, and it's also very large since it has this group over here. And so that would also make it hard, like it makes it inflexible when we have it in a polypeptide chain. All right, and then this is kind of like a summary between like what R group interactions involved with tertiary and quaternary structure. Again, like I mentioned before, it could be ionic bonds, and an example here is lysine, which is positively charged amino acid, and aspartic acid, which is negatively charged. When we have 2 fully charged atoms, they interact, and their opposites interact, and they would form an ionic bond. We could also have interactions such as ion dipole interactions, and the example here it would be lysine, which is 1 charged amino acid, and then the serine, which is not charged. That would make an ion dipole since we have a 1 fully charged atom and 1 amino acid, and then 1 like partially charged atom. We could also have non-polar interactions, such as van der Waals or London dispersions. And we could have hydrogen bonds. And again, that forms between a hydrogen that covalently bonded to a nitrogen, oxygen, or fluorine. And then that hydrogen would interact with another oxygen that is covalently bonded to another molecule or a nitrogen or a fluorine. But like, we can't have a carbon that is covalently bonded to a hydrogen, and we can't take that hydrogen and make an interaction with an oxygen. Does that make sense? Because it could be a little confusing. Okay. And then we could also, yeah, the hydrophobic would make the van der Waals or London dispersion. And for the hydrophobic effect, that's where in the amino acids, the way it folds, we're going to have all of the non-polar amino acids fold kind of like on the inside, or that's the way like it's preferred, and the inside of the protein, so it's not exposed to water and like to not aggregate. And this is another just bond summary for each structure. So for primary structure, we only have covalent bonds, peptide bonds, which are covalent. And then for secondary, we have hydrogen bonds, but it's only between backbone atoms. It doesn't involve the R groups. And for tertiary and quaternary structure, we just went over it. It involves both covalent and non-covalent. Okay, then I just have a few questions for that section. First one is like, which of the following amino acids would be bound to an amino acid with positively charged R group in a tertiary structure? Feel free to type on the chat or yell it out if you're in the room here. Anyone?
Expectations were so low for final exam grades. Maybe that's for this. Just kidding.
I don't
B?
know. B.
Say B?
Yeah. Why? Exactly. If people in Zoom didn't hear it, we said that B would be the right answer, and that's because glutamic acid is a negatively charged R group or amino acid R group, and it would interact with a positively charged R group, such as lysine, and that would form an ionic bond.
Did you have something to add or question?
So already running on the type of polar, not polar, hydropolar, and technical, polar, non-polar, positive, neutral, negative, neutral, like amino acid choice. Or will I go to find out next or not?
The amino acid chart would be provided,
yeah.
but like in terms of which one is like more
Well,
or less.
Yeah, I think
Yeah,
you will
the
have
chart-
this
yeah.
chart, this
Yeah.
exact one. Yeah.
But for hydrophobic to hydrophilic, I would write like the list that's in like in one of those exams, or sorry, not exam, in the lecture
Debate.
video for that week. Because like, I mean, I can't tell you what's in the exam, but I mean, it's not impossible that there'd be a question asking you which one's more hydrophobic, giving you two of them. So even if like you're on the exam, you're like stumped of like, I don't know why, you at least can get the points if you just write down the list. It barely takes any space on your like 3 like note sheets that you can bring. Mhm.
Okay. I have a second question, which is like, we kind of talked about it, but alpha helices or beta sheets are found in every protein. We don't necessarily have to have both of them in each protein, but we could have either or, and some proteins have both structures. But what characteristics of their bonding makes it possible that it will be found in every protein? Yeah.
That way.
Outside.
Yeah, I'd probably walk outside and then head down the street. And then if you go, you know where like the stairs are?
Yeah.
What room and moral are you looking for? 375?
I'm not sure. It's not like a ballot discussion. A bar review. It's like I just saw 4.
Yeah, go all the way up the stairs and then take a left and you'll see the entrance for,
all the way left.
yeah. Go down the street.
The stairs,
Go up the
it's
stairs.
the one on the left.
Okay, thank you. You got it. Sorry. Thank
Good
you.
luck.
Okay, does anyone have an answer for this one, or do you guys just want me to go over it? Okay. I'll just go over it. Okay, so we said that the type of interactions that take place for secondary structures of alpha helices and beta sheets is hydrogen bonding between backbone atoms. So it does not involve R groups. And we know that R groups are what give characteristic or like uniqueness to each protein. So since they're not involved in that level, that means that the secondary structure is not unique to a specific protein, or we don't have like a specific secondary structure for every protein. That's why they could be found in all protein. And the answer is right here. It didn't working. Okay, here we go. Does that make sense, or do you guys want to? Okay, great. One more question. True or false? The amino acid proline is not found in alpha helices because its R group is too large to fit into the alpha helix. It just what do you think? True or false? Some could be tricky, but anyone in the chat? Why? Yeah, so it is true that proline is not found in alpha helix, but it's not for the reason that it is too large. As you said, it's not too large compared to other. Yeah.
Is it because like the alpha helices, the R groups like stick out anyway? They're not like constrained to the middle of the helix.
Yeah, that's part of it. It doesn't have to be like our groups are not involved, they're gonna be out anyway. But the reason that the proline is not involved in alpha helices is because it's connected, like the R group is connected to both the central and the amino group, as I showed you before. Wait. Yeah, right here. We can see that it's connected to, it's just 3 carbons, as you said, just small. But it's connected to both the central and the alpha amino group, which forms like a cyclic shape. And this is this makes it form like a unique kink shape that makes it inflexible to form or go around with like the way the helix would form. Okay. I think that was all it for the question. And for like topic 1, does anyone have any questions on that section? Any question? Okay, if questions come up, just feel free to ask. Now we're gonna go on protein unfolding and misfolding. Okay, so we know that environmental factors have an effect on the protein structure and therefore the function of the protein. And some of these factors are temperature, pH and salt concentration, and light energy. And the cell can regulate these environmental factors such as the pH, the temperature, and the salt concentration. Doesn't necessarily like control light, because that would be like an outside factor. But if we have a protein that is misfolded, that means it's denatured and it's abnormal. When we have it misfolded, that means we're changing its structure or its shape. And we know for proteins, once we change structure, that means we're changing the function. So when a protein misfolds, it does not do its proper function that we need it to do for the cell. And the cell has some ways to fix these unfolds. And we're going to talk about these later on the slides. Okay, so how are the bonds affected? It causes disruptions to all, for this class. We're gonna, like, when we talk about disruptions to bonds, we're gonna talk about like disruption to hydrogen bonds, electrostatic interactions, all kinds of bonds that are non-covalent. Covalent bonds are not affected in this class when we talk about like disruptions that happen because of environmental factors. And that's because here in this class, we say that covalent bonds are very strong and they're gonna need energy, a lot of energy and like specific enzymes to be broken. Okay. Okay, so the way heat or thermal energy would attack the non-covalent bonds, it would cause it to denature or unfold itself. We're gonna have like the ball peptide unfolded. And an example here is like if we're cooking a scrambled egg, we're gonna have it change shape once we blow down the heat. However, we say that covalent bonds are not affected, or the primary structure of the protein is not affected, because once you eat the egg, even if it's scrambled and it lost its like original structure, you're still getting a protein from the egg. So that means that the primary structure of the egg is still intact. And usually, like we say in general, that if we have less heat, it's more stable, and if we have more heat, it's less stable. But more stable is not always better for the protein. It depends on the function of the protein and like what conditions it works under. So for like human proteins, they work best under 37 degrees Celsius. For other organisms, it could be higher or could be lower. Okay, and for pH and salt concentration, the ways it could affect it is they would affect the charge of the amino acid R groups. And if we affect the charge of the amino acid R groups, that would affect the bonding that is taking place. So let's say we have a lysine and it's making an interaction under a pH of 7. So it will still have its positive charge and it could form an ionic bond. But let's say the pH goes up very high, above like goes to 11. This will take the positive charge away from the amino group of lysine, and it wouldn't be able to make an ionic interaction that it did before. And then the last type that we could affect protein is using light energy. So high wavelengths would also disrupt the non-covalent bonds and could break them or unfold the protein in the same ways that for other ones we talked about. Yep. Do you guys have any questions so far about any of the stuff we talked about? Okay. Great.
So the next part we'll talk about in topic 2 is how you would go about resolving a misfolded protein, or how the cell would. So the key points to remember from this kind of review is that cells have a variety of robust mechanisms that help with the problem of protein misfolding. And so proteins that are misfolded in the cytosol or in the nucleus are sent to the proteasome in a pathway called heat shock response, or HSR. And for proteins that go into the endomembrane system, there's a specific ER stress response called unfolded protein response, which is also called UPR. And a lot of neurodegenerative diseases are caused by misfolded proteins or aggregations of them. So like mad cow disease is like a disease where if there is a protein that's misfolded and then it touches or it comes near a protein that is normally folded, it can cause that one to then misfold as well, and like form aggregate clumps where they are insoluble because the the cell will unravel and the hydrophobic core becomes exposed. So there's these proteins called chaperone proteins that help to recruit other proteins fold or stabilize proteins when they're unfolded or misfolded. So it's most likely that the chaperone proteins are the ones that are going about and sensing the misfolding or unfolding. And this is kind of shown up here in this diagram. And it's good to think of these like a chaperone on a field trip. When they go on the field trip, they're there to supervise the children if they get out of control. So if the proteins get out of control, in a sense, like becoming unfolded or just misfolding, they can help with this. And one thing to keep in mind is like how this fits in the big picture, like unfolding, the process of unfolding in like here, how does like something get to this from being folded to unfolded? That could be through a variety of mechanisms, including like heat, which then would degrade like quaternary, tertiary, and secondary structures and just leave it at primary. So heat shock response is a process that occurs in cytosol in the nucleus. And the image below shows this process going on. And basically, these proteins are tagged by ubiquitin, and then it gets degraded in the proteasome through ubiquitin-mediated degradation. So it's sent to the cells kind of like trash disposal. And unfolded protein response is a different response that in which the cells sense unfolded proteins in the ER. And it's a bit more complex of a system. So when it initiates this unfolded protein response, translation decreases in the cell to reduce the folding load into the ER, because the cell wants to focus on the problem at hand of unfolded proteins before it makes even more. And then it also increases chaperone production, because chaperones are proteins as well. And the cell needs more chaperones to help to refold these proteins correctly. And then another thing that happens is that lipid synthesis increases. So this will increase the maximum volume of the ER to accommodate for the increased volume of proteins inside the cell, mostly chaperones. And then how does the ER get rid of this or these bad misfolded proteins? If it can't be refolded, then it has to go. And since there's no proteasome inside the ER, unlike the cytosol, the protein has to be retrotranslocated out of the ER so that it can be sent to the proteasome. And like this is one of the only times that you'll see this occurring, that when a protein goes into the ER, because as you'll see like in later sections, like once a protein goes in the endomembrane system, it usually doesn't come back out, but this is an exception to that rule. And here's just a diagram showing how this is occurring in the ER. Antonio, take it away.
All right, so now we will talk about protein ligand interactions and protein regulations. So protein regulation is kind of under three main points, which are allostery, post-translational modification, and nucleotide binding and hydrolysis. So we're going to go into depth in the next slides into each of these. So to start with allostery, it's a concept that essentially means protein function is regulated by its shape. So its structure essentially equals function. So another part of allostery is allosteric regulators. And so ligands bind to active sites of proteins in order to initiate some sort of response. But allosteric regulators refer to ligands that bind somewhere other than the active site. And this can change the activity of a certain enzyme, and it could either be activating or inhibiting. So on the bottom left hand side with the green ligand, you can see that it's binding not to the active site of the yellow protein, but that binding causes a formation of an active site, allowing that blue ligand to now bind to this yellow protein a lot easier. And then on the other hand, with the red ligand, it's binding somewhere other than the active site, which is closing the main active site for the blue protein, inhibiting its function. And for post-translational modification, this refers to when proteins are chemically modified after they are made. And this usually happens on a specific amino acid. And this can change the surface chemistry of certain proteins, and this can cause a conformational change, and therefore changing what it interacts with and what it does. And some examples of post-translational modification are phosphorylation, and that's addition of a phosphate group. This little mechanism is shown here on the right side. And ubiquitination, and that's the addition of ubiquitin chain on a protein, and these marked proteins are usually sent to the proteasome to be degraded. And lastly, nucleotide binding hydrolysis. ATPases and GTPases are proteins that are categorized as enzymes since they can bind to ATP or GTP and hydrolyze them to ADP or GDP. And this mechanism is just by breaking the bond between the second and third phosphate. And this is changing it from a 3-phosphate chain to a 2-phosphate chain. And RAS is a example of a GTPase, and its little mechanism is shown down at the bottom. And then for KD, its equation is given on the left. I don't think you need to actually ever calculate KD, but just so you guys know, the KD is equal to concentration of protein times the concentration of ligand, all divided by the complex, which is just the ligand and protein concentration together. And I think a good thing to just know is that KD is directly or indirectly related to affinity. So if you have a high KD, you have a really low affinity, and vice versa. If you have a low KD, you have a really high affinity. And now we will move on to enzymes. Does anyone have any questions about post-translational modification, protein regulation, anything like that? Anyone in the chat? Okay. So now we'll move on to enzymes. Cells need to make and break covalent bonds quite often, and enzymes are truly necessary for this to happen because making and breaking covalent bonds are very difficult and can take a very long time. So enzymes really speed up this process dramatically. And they're very heavily regulated in cells, so this gives cells very good control on what kind of reactions are happening. How they work is they essentially lower the activation energy for certain reactions. So the activation energy is the amount of energy needed to fulfill a reaction. Therefore, an enzyme lowering it means less energy is needed for the reaction to occur, therefore going faster. But make sure you know that enzymes are only affecting the activation energy and not the energy of products or reactants or their concentrations. And question for you guys, is the diagram on the bottom right of the screen endergonic or exergonic? Okay, well, it's exergonic because it's releasing energy. Here you can see that the energy is lower in the products, therefore some energy was released into its environment or its system. So for enzyme affinity, their affinity changes based on if it's in the presence of its substrate or product. Our enzymes really want a high affinity for their substrates because they want to make sure that reaction goes as fast as possible. Therefore, it's going to have a really good affinity for that substrate to hold on to it, make sure that reaction is being fulfilled. But once that reaction is done, you don't want a high affinity for your product anymore because you want that product to go out into the cell and do what it needs to do. So therefore, enzymes want really low affinity for their products. So just remember that it's high affinity for substrates, low affinity for its products. And that was it for topic 2. Does anyone have questions on anything so far before we go on to topic 3? If not, then we'll move on. So now we're going to talk about protein trafficking and gene expression. So proteins are needed to be trafficked to specific sites inside or outside of the cell in order to function properly. And some examples of those are like insulin. It needs to be released into the bloodstream, even though it's made in a cell. It needs to somehow get out of that cell into the bloodstream. And as well as DNA polymerase, it's made somewhere in the cell and it needs to make its way into the nucleus in order to function. And for eukaryotic cells, we use signal sequences in order to localize these proteins to specific sites. So ribosome location is a really important determination of where proteins are going to be localized. So free ribosomes that are just kind of floating in the cytosol, these make proteins that are normally not bound for the endomembrane system. There's a mistake on this slide, my bad. Exceptions should not be on there. Yeah. It's okay. Just don't disregard the exceptions. That's not supposed to be there. So free ribosomes are going to make proteins that are not bound for the endomembrane system, and these proteins are normally localized to either the nucleus, mitochondria, peroxisomes, or chloroplasts. And then on the other hand, ER-bound ribosomes are usually making the proteins that are bound for the endomembrane system. So a question for you all. What parts of the cell are included in the endomembrane system?
The ER, the Golgi apparatus, and sort of well, like pretty much like okay, well, the Golgi and like the ER. So once the ER, it shipped like I just modified shipped out to the Golgi, which shipped it out to the rest of the system.
Mm-hmm. Yeah, you're right. So it's the ER, the Golgi. Lysosome is also a part of the ER, and the cell membrane, because through the endomembrane system, that's how proteins are embedded in the cell membrane, as well as the transport vesicle that's used to move between like the ER and the Golgi and the cell membrane. So if any protein needs to enter that endomembrane system, it needs to have an ER localization sequence. And if any protein needs to function inside of either the ER, Golgi, or lysosome, it's going to need some sort of retention sequence, because without that, it's just going to get kind of spit out of the cell. And then this is a little picture of the main like movement of the endomembrane system. So now signal sequences. They're they're essentially just a group of amino acids within an amino acid chain that tells a protein where its destination is going to be. It's part of the primary structure, so it's not added after anything's made. It's actually embedded inside of the actual amino acid sequence itself. And localization and retention sequences. So going back to what we said about the endomembrane system, ER localization sequences are needed for any protein that needs to be sent to the endoplasmic reticulum or the endomembrane system. And in order to stay anywhere along that point, it's going to need some sort of retention sequence. And then for the nucleus, needs nuclear localization. And for mitochondria, same thing, needs a mitochondria localization. So for ER localization, once the protein has that sequence, it's going to be bound into the endomembrane system, and it's going to stay in that system until it's secreted. This is kind of just the same information. It needs to stop anywhere. It's going to need some sort of retention sequence. And this is a little flow chart. I know it's kind of daunting to look at, but it's actually quite helpful to look at. Maybe you could put this on your sheet just to remind yourself where everything's going. And this is a little summary on everything. I'm not going to read through this. We'll send you the slides. But it's a nice little summary if you want to put this also on your note sheet, just to remember where what everything does. And a question for you guys: if a protein is made to replace a receptor on the cell membrane, this protein was translated by a ribosome that's bound to the ER, and imagine that this protein underwent some sort of mutation where the retention sequence was deleted. Assuming that the protein's ER localization sequence is still intact, where will the protein end up after it's made? And a hint is, what do retention sequences do to proteins in the endomembrane system? Yeah. Basically, yeah. If it doesn't have that retention sequence, it's just gonna end up going through the whole thing and get secreted. And now nuclear import. So there's a lot of steps to this, but I believe you'll get the diagram for the exam. So essentially, we'll just run through it real quick. Starting off here at number 1 at the bottom right corner, important and its cargo protein are going to come together and bind and enter the nucleus through some nuclear pore. And once it's inside of the nucleus, RAN that is bound to GDP with the help of a GEF will facilitate its change to a GTP bound state. And this allows that RAN to bind to the cargo protein and important complex. And once that complex is formed, there's going to be some sort of conformational change that releases the cargo protein. And now that cargo protein can work and function inside of the nucleus. And then that GTP bound RAN complex with important is now able to exit the nucleus. And once outside back into the cytosol, it will, through the help of a gap is going to bind to RAN and increase its ability or efficiency to hydrolyze GTP to GDP. And once it's bound to GDP, it will release important and the cycle will just start again. And it's important to remember that the cargo protein does have a NLS sequence on it. And a good thing to remember is GEFs are normally found in the nucleus and GAPs are found in the cytosol. And then now on nuclear export, it's kind of very similar. We're starting in the nucleus with exportin and a cargo protein. So when those have to leave the nucleus, RAN is going to go through the same process with RAN-GEP, allowing for that exchange of GDP for GTP. And once it's bound to GTP, that complex of RAN, exportin, and its cargo protein are all able to now bind together. And also notice that the cargo protein has a nuclear export sequence. So once that complex is formed, it's able to be released into the cytosol. And once in the cytosol, another gap again will come and bind to RAN, increase its ability to hydrolyze GTP for GDP. And once it's bound to GDP, the cargo protein as well as exportin will be released into the cytosol, and exportin will make its way back into the nucleus where it's supposed to function. That was a lot. Does anyone have questions about import or export? We'll go over it. So GEFs and GAPs, they're general terms for the RAS family of GTPases. So GEFs are the abbreviation is guanine nucleotide exchange factor, and this facilitates the change or the exchange of GDP for GTP. So once it's bound to RAN, which is a GTPase, it's going to initiate some sort of conformational change, which is going to decrease its affinity of GDP and allow it to be released. Therefore, GTP now has a pocket to bind into. And for GAPs, these are GTPase activating proteins. So they stimulate the intrinsic ability or intrinsic activity of RAN. So once it's bound to RAN, RAN is able to hydrolyze GTP into GDP much faster than it normally would. And for you guys, how would nuclear export be affected if there was a loss of function mutation in RAN gap? So, oh, sorry, go ahead. Yeah, essentially. So if RAN gap is no longer functioning, it's not going to be able to bind to RAN and increase its ability to hydrolyze GTP anymore. So RAN's going to take a very long time to hydrolyze that GTP into GDP, and it's going to stay bound in that complex at number 4 a lot longer than it normally would. So therefore, over time, nuclear export would stop because exportin will start to accumulate in the cytosol and not be able to re-enter the nucleus to continue its process. So for transcriptional regulation, there's a few different factors. Transcription factors regulate DNA polymerase's binding to a promoter on DNA. And in eukaryotes, we have basal transcription factors, and prokaryotes, we have sigma factors. And essentially, positive transcription factors upregulate transcription and negative suppress transcription. And a eukaryotic example would be heterochromatin versus euchromatin. So chromatin is where how DNA is packaged very tightly, or sorry, chromatin determines how genes can be expressed. So euchromatin is the loosely packed form of DNA, which is shown at the bottom. In that loosely packed state, our RNA polymerase is able to come in and transcribe and transcription factors are able to access the DNA to transcribe or express some sort of gene. Versus heterochromatin, which is shown at the top, where this DNA and histones are very tightly wound together. Therefore, RNA polymerase and transcription factors cannot access the DNA and won't be expressed or genes won't be expressed. So now going on to RNA processing in eukaryotes. Once RNA is made, a 5' guanine cap can be added to the 5' end of RNA. And this prevents any chewing from nucleases that could be floating around in the cell. And another one is a poly-A tail, and it's a long chain of adenine nucleotides, which are added onto the end of some mRNA strand. And that strand is able to get chewed over time, and it's a non-coding region. Therefore, the actual important coding part will be protected from any degradation. And it's also required for export from this. And further RNA processing in eukaryotes, also known as splicing. So introns are the areas of non-coding material in DNA that are RNA, that are able to be spliced out. Therefore, leaving the exons, which are coding regions and are necessary for making some sort of protein. And spliceosome is the enzyme that is responsible for trimming out those introns. And there's also alternative splicing, which allows for multiple different proteins to be created from the same stretch of RNA. Shown at the bottom, one way it could splice out the green exon as well as the introns, which are the black lines, to create some red, yellow, and blue protein, or mRNA, which creates some different protein. And then it could also take out the yellow exon to create some other type of protein. And this is just the little flow chart of how gene expression is regulated in eukaryotes. And I would say a good rule of thumb is just to name one or describe and name an example of each thing on this chart for the exam. I think that would be helpful. And then this is just a little summary slide of prokaryotes and eukaryotes, differences in how they regulate their gene expression. I'm not going to go over this because we just did pretty much all of it. So these slides will be sent out, and now we'll move on to Gates.
Yeah, so the last stop on our crazy, wild, but amazing adventure today in this winter wonderland of topics 1 through 4 is going to be the regulation of genes through, or gene expression through a couple mechanisms like the trip operon and microRNAs. So the first part that we'll talk about, which was talked about a little bit before, is basically just transcription factor mediated transcriptional regulation, which basically just is about how transcription factors control how genes are expressed by allowing by binding to promoters in genes so that the RNA polymerase can come by and transcribe them. And so general transcription factors include sigma factors in prokaryotes, basal and eukaryotes. And then, as discussed before, regulatory ones are specific, and there are positive and negative ones. Positive transcription factors, when they bind to DNA, cause an enhancement of transcription of the genes, whereas negative ones will suppress the transcription of these genes. So a sigma factor that we talk about is sigma 70, and it's a protein that helps RNA polymerase to bind to the promoter. So it's a positive regulator, and sigma 70 is sequence specific, but it's not an exact sequence. There's a general consensus sequence for this sigma, which means that there can be like slight variations in this box that it binds to between 35 and 10 base pairs in the promoter behind where the mRNA starts, or where the DNA that holds the information for the mRNA starts. And so there's a bunch of them that can match up to it, but the best that works for is like this general consensus sequence. And because of this, it can regulate, because it can vary slightly, it can regulate a lot of genes. A single one at least can, and it can bind to various varieties of these sequences. So affinity will change depending on how much this sequence in the DNA of the promoter varies from gene to gene. And there's some outdated mean. And then, so in terms of operons, an operon is a series or length of DNA where it contains regulatory DNA sequences that act as a place for binding of regulatory proteins. And the operon includes two sections. One is the promoter, and this is where the RNA polymerase is going to bind. And the operator is where a repressor can potentially bind to the DNA in order to prevent the RNA polymerase from binding and transcribing the DNA that comes after the operator. And operons can either be inducible or repressible. An inducible operon is usually off by default, but it can be turned on in the presence of a specific molecule, or it can be usually on, but turned off in the presence of a specific molecule. So repressible is the example that we focus a lot on in the example of the trip operon. So in the trip operon, there's a few different scenarios that we're working with. The first scenario that we'll focus on is low tryptophan levels. And in low tryptophan levels, a bacteria doesn't have access to tryptophan. And tryptophan is an amino acid that they need to make proteins. So trip is not, the trip operon doesn't code for tryptophan. It codes for proteins that help make tryptophan. That's a little bit of a common misconception. And so when these genes are on, in the cases of low tryptophan levels, the proteins will be expressed from this operon that will come together to make tryptophan that's needed in the cell. And in cases of high tryptophan levels, bacteria will have a lot of access to tryptophan, so they won't make the proteins to produce tryptophan. They'll be inhibited by the repressor, and we'll show these in the next slides, but what is the question? The TRP repressor protein is a negative regulator because when it's active, it causes the operon not to be expressed. So here's a TRP operon diagram. Yeah, so in the TRP operon here, you can see that there at the beginning of the operon, well, this is the TRP operon itself, all of this downstream of TRPR. And so TRPR is a gene upstream of the TRP operon that codes for the repressor that works on the TRP operon. And so by default, when there's no tryptophan in the cell or low levels, then the tryptophan repressor won't be active, and so it will allow for this transcription to happen as it's not bound to the operator. The same thing can happen in medium levels of tryptophans in the cell. If there's not enough for to bind to the repressors to activate them to go in the operator, that leads to attenuation, which is controlled through this attenuator sequence, and that'll be shown in the next couple of slides as well. And so, yeah, in low levels of tryptophan, they won't bind. In high levels, what happens is that tryptophan will end up binding to the repressor, and that'll make it active. And then when the repressor is active, it will bind to the operator region shown here. And when it binds to the when the repressor binds to the operator region, then that doesn't allow RNA polymerase to bind to the DNA to transcribe the rest of the genes. So in this case, attenuation can't even happen because attenuation is a mechanism used when there's enough tryptophan in the cell that it doesn't need to, or it shouldn't create proteins to make tryptophan, but there's not quite enough to trigger this mechanism of binding to repressors to bind to the operator. So there's no initiation in this case of high tryptophan where it's binding to the operator. There's no initiation of transcription at all. So now we'll go over attenuation, which is a regulatory system used when there is a medium-ish level amount of tryptophan in the cell. So attenuation is the early termination of transcription, which means that it can stop the transcription of the rest of the structural genes in the operon for tryptophan. And this is before the DNA sequence for TRP proteins. And there's 4 regions that are shown here. And these 4 regions are complementary to each other. There's a few different pairs that can form. There's a pair between regions 1 and 2, and 2 and 3, and 3 and 4. And so the main reason why they can do this, the prokaryotes, is because they have transcription and translation happening simultaneously, unlike in eukaryotes where transcription is happening in the nucleus exclusively, and then translation's happening outside of the nucleus. Oh, right. And so, yeah. So when 1 and 2 are, when 1 and 2 and 3 and 4 hairpin, this is going to cause the termination. I'll go more in depth here. So, oh yeah, we already went over this low level. So RNA polymerase is going along these regions here. And if it gets stuck on these, there's trip operons shown in this diagram over here. And so it goes along, and if it stops, it's going to stop there if there isn't enough trip operon or tryptophan in the cell. And if that happens, then the ribosome is going to get stuck on region 1, and this is going to allow regions 2 and 3 to hairpin and like bind to each other. And when that happens, the RNA polymerase will need to, or sorry, the RNA polymerase, will wait until the ribosome gets stuck, and then RNA polymerase can continue transcribing region 3. Then it'll form this hairpin. And if 2 and 3 hairpin here, then that means that attenuation won't occur, and the transcription and translation of the entire mRNA will occur, including the later structural genes. So in a different case where there's not enough tryptophan, or sorry, where there is enough tryptophan in the cell to trigger attenuation, but not enough to stop the initiation of transcription, attenuation is going to need to occur. And the way that happens is that RNA polymerase is going to go through these 1 and 2 regions, and the ribosome is not going to stop on these tryptophan coding regions, because there's going to be enough tryptophan in the cell to be translated here. Because remember, the ribosome is it's attached to the mRNA coming out of the RNA polymerase that it just transcribed from this DNA region. And so that mRNA has codons that code for tryptophan. And so it's going to like request tryptophan. And if that is able to happen, then this process is going to go on too quickly. And the ribosome is going to get stuck on the stop codon, like right before region 2. And then this is going to allow for the hairpin to form between 3 and 4, because the ribosome is on top of 2, which prevents 2 and 3 from forming. And if 3 and 4 hairpin, then this is going to cause the termination of this transcription, and RNA polymerase will dissociate, and this whole mechanism is going to stop. So here's an overall summary of what's going to happen with the trip operon. And you guys can just use this to review. We just went over this. So the next topic we'll go over is RNA interference. And here's just like a summary diagram. Oh, yeah.
All right, thank you.
Are there any other questions on this attenuation mechanism? I know it can be a little difficult, yeah.
Like, how does, trip versus trip tRNA levels like play into attenuation? Because like I had written down, I think in my notes, it was like attenuation occurs when there's not enough trip to suppress the start of transcription, which I get, but then it's but enough trip tRNA to do translation. I don't get that.
To do, yeah, so there's an amount where of tryptophan where there's not quite enough. There's not quite enough to a bind where there's enough to bind to the repressor. That is on this slide over here, right? So if there's not enough to bind to the repressor, like they're not high enough concentrations where they can like bump into each other in order to bind, then what's going to happen is that this repressor is not going to be able to bind to the operator, but then it's going to like continue on obviously into attenuation. RNA polymerase will continue to transcribe, but then it's going to reach a point, RNA polymerase, where it's like going down the mRNA after like this region, and then it's going to reach this region over here where it just transcribed the DNA, which codes for 2 trip codons. And then at that point, once that has been transcribed and there's like a mRNA like transcript there, the ribosome is like following behind it. And when the ribosome gets to that, because there's a bunch of along the entirety of this transcript, there's a bunch of codons, each like coding for a particular amino acid. And then eventually the ribosome is going to get to this amino acid, which happens to code for tryptophan. And so like the way it works is that like, it'll signal for, okay, need tryptophan, like the ribosome will, and then the tRNA that's like floating around the cell and like attached to the tryptophan will then like bring over a tryptophan, and it will bring over another, then there'll be another one because there's 2 right here, and then it'll just continue along in that process of having like requesting for tRNAs specific to a amino acid to like bring over that amino acid. So in this scenario, we're saying that there's not enough to like stop initiation of transcription, but there's enough that you can have that there will be tRNAs that will come like quickly enough with the trip amino acids. Does that make sense, or does that clear it up? Okay. Any other questions? Any on Zoom? Okay. So the next thing we'll talk about is RNA interference. So this is just like an overall diagram that just goes over both mechanisms that we'll cover. So the main ideas of RNA interference is that the cell wants to either degrade or suppress pieces of mRNA in the different areas of the cell, and it wants to suppress like their translation. They've already been transcribed, but now it wants to put a stop to that. translation into the proteins as a way to just regulate overall levels of these of the expression of these proteins. And there's a few main components of this. There's risk, which is involved in both pathways that we'll talk about, and it stands for RNA-induced silencing complex. And it's composed of two different parts. There's the risk itself, and then there's Argonaut, which is sometimes abbreviated as AGO in a lot of the diagrams that you'll see. And Argonaut is the actual enzymatic part of the complex that is responsible for degrading mRNA. And then risk is the protein like holding it and binding to, so that this complex will bind to a small guide RNA that uses this guide RNA to target the mRNAs that it wants to suppress the expression of, using some sort of base pairing complementary system that varies between the two slightly. RNA can form higher order structures and folding in on itself. And we call these stem loop or hairpins. And these hairpin structures, pre-RNA, are substrates for drosha to cleave into smaller pieces, which we'll see in the next diagrams. siRNA, and then we'll move on to these two right here. These two different types of RNAs will be suppressed in different ways because they have different origins. But their overall mechanisms are similar, as you'll see. And so siRNAs are small interfering RNAs, and they usually come from exogenous sources like viruses that invade us. And they're cleaved by dicer in the cytoplasm, and it usually leads to mRNA degradation. And microRNAs or miRNAs come from endogenous sources, like inside of our cell, and they must be translated and transcribed from our genome in the nucleus. And most of the times, this pathway leads to translational repression, but it can also lead to mRNA degradation, which also leads to translational repression. So for miRNA, this is the overall diagram. And so the pathway of mRNA is that usually when, actually, I think it's better. Do you guys prefer if I just show this diagram and then like tell you this? Or do you guys want to see this while I'm talking? Diagram, words. Diagram, words. Which one? Diagram, okay. So when initially transcribed in the nucleus, the RNA is called pre-RNA with an I. This is mislabeled. In the nucleus, drosha will process and cleave the pre-miRNA into smaller pieces called pre-miRNA. So it is correctly labeled, but on the second one here. And pre-miRNA will exit the nucleus. Keep in mind with the help of exportin, with the diagram that we talked about before. And then dicer, which is like a pair of scissors, in the cytoplasm will process and chop up this pre-miRNA into smaller pieces called miRNA duplexes, as you can see here. But these are still double-stranded, and then RISC it'll bind to the miRNA and it'll unwind these two strands, and then it'll use one as the guide strand. And keep in mind, this is a single strand RNA, and then the other strand gets degraded. And then the RISC complex, which is now attached to the guide strand, can target mRNAs and cause degradation or translational repression. And so RISC mRNA uses base pairing complementary to target certain mRNAs. If it's a perfect match with the mRNA, it'll lead to degradation through Argonaut. However, a lot of the time it's imperfect or partially perfect binding. Not all like the base pairs might exactly match, but this will end up leading to the RNA being stored in the P bodies and in this part right here. So if it's an imperfect target, it'll send the mRNA target to the P body for decay or storage. And then siRNA, there's a better diagram on the side, honestly. So for siRNA, this pathway is pretty similar, but it's got some differences as well. It also starts in the cytoplasm with double-stranded RNA or shRNA, and it's cleaved by dicer. And then RISC will unwind this duplex right here, and it'll pick a guide strand with the help of Argonaut. And then this RISC siRNA complex will form. And then this will be used to base pair with target mRNAs. And unlike in miRNA pathway, there's no option for imperfect matches. In this case, it's going to be an exact base pair matching of the mRNAs. And then Argonaut will degrade or silence the mRNAs. And one thing to keep in mind is that one siRNA guide strand can cleave many target mRNAs. It's just that they have to be of an exact sequence. So why doesn't RISC grab just a single strand of RNA? If it needs only one, why doesn't it just grab one? Single-stranded RNA tends to be more unstable than double-stranded, and it's more quickly degraded as compared to double-stranded. And so it won't really be able to interact with RISC. And the process of RISC choosing the guide strand is vital to the pathways of RNA interference, which means it has to select one and then degrade the other. It wouldn't recognize a single strand of RNA. The way it normally operates is to make this choice between the two. The strand that it chooses is called the guide strand, and the strand that's not chosen, it gets discarded, is called the passenger strand, but I don't think you need to know that one. You do need to know what guide strand means though. And then RNA silencing cannot work properly if the mechanism doesn't occur as normal, and normal being double-stranded RNA, to do its job. Are there any questions on those before I move on to the similarities and differences between the two? Okay, so miRNA and siRNA have different sources. And so the way I like to think about it is, so one comes from within our genome, one's from outside. The way I like to think about it is miRNA is my RNA, so it comes from within our own genome. Whereas siRNA, I have another acronym for it, but it's a lot more complicated. So we'll just stick to this for now. So then miRNA starts in the nucleus, and it has dicer processing and it has DROSHA processing. Whereas siRNA only has dicer processing. And then also with mRNA and siRNA, a big difference, like I was just talking about before, is that with mRNA, it can be, and it usually is, an imperfect base pairing with the target mRNA. And if it is imperfect, then the mRNA will be sequestered into the P body. And this usually leads to like transitional repression. But in cases where it's a perfect base pair match, then it can also lead to direct mRNA degradation. Whereas with siRNA, it has to be a specific mRNA. And in both, it can target multiple different mRNA strands. It's just that in mRNA, it can target a variety, like a larger variety, because it can not only do multiple mRNAs, but also multiple varieties of mRNAs with different sequences. And that is the end of our part one of this thrilling series of your favorite exam coming up this semester. Are there any questions? Content-wise? Yeah. Oh.
So we could write out, you're saying like the hydrophobicity of the amino acids or the R groups that you were saying?
Yeah. Wait, sorry,
Like-
what's your
Like
question?
you were saying that our like cheat sheet, we should write out
Yes,
like the order of
yeah,
is there somewhere like you can find that? Or-
yes, yeah. If you go to, I mean, I can like send an email with it, but actually I can't. Our email thing is really weird, but if you go to like in the first week, if you go to like one of the videos, if you just like scroll through, there's a slide that just shows like straight up, like it's just like hydrophobic at the top and like hydrophilic at the bottom or vice versa. And it's just a straight up list of them from one side to the other. So I mean, it would barely take any space on your page to just write all the words. Yeah. Also one thing we didn't mention, but I'll mention it now for people watching the recording as well, is that your lowest exam grade will be dropped if you scored better on the final. So if you really, really bombed one of the first two exams, it would be very much in your best interest to study very hard for the final. Also, you can bring two sheets, like the two cheat sheets from the previous exams and an additional one, though it's not, she's not looking to see that it's the exact as the same as the previous one. So the rule is just that like you can bring 3 cheat sheets. So I just recommend that like the first one be like from the first exam, the second one from the second exam, and then like third being from like the material from then on out, plus whatever material you want to like add in. And I would definitely recommend like hand writing them if you have the time, because it really helps to like ingrain these ideas like in your head, because it's, even if you don't end up like looking at it, it's good to just like write this stuff down. So yeah, I'm just gonna end the recording here. Oh, sorry. Yeah. Are there any other questions on Zoom or in person? Okay, thank you everybody for coming. And I'm going to end this meeting.